Okay, so this is chapter 13, and we're going to talk for a little bit about meiosis. And if you guys printed out the note handout that I sent you, I'm just going to follow along that note outline. So sort of an introduction to the chapter, I usually show this picture because it reminds all of us why organisms do meiosis. And the main reason, of course, is this is going to happen only in organisms that are diploid, they're 2N, and only for sexual reproduction. That's the whole purpose of it, is to make gametes, right? Which is another name for sex cells. Now, in humans, of course, the sex cells are, we call them eggs and sperm. And you can see we've got male and female sexes are separate, and we have a special organ and tissues to produce those sex cells. In other organisms, it isn't always like that. So I put these slides in to show you some differences in other organisms. But after I typed all that out and included them, I thought about it a little bit. And if we were having this in class, I usually just ask you definitions about those other slides. So I'm going to focus on this slide for a few minutes, and then we'll quickly go through the next 3 slides and try to remember that for those slides, it'll just mostly be definitions on the exam. So as I said, here we've got sexes are separate in humans, and we have specialized organs to produce the sex cells or the gametes. And when the gametes fuse, so when the sperm meets an egg, that's when we have fertilization and we form a diploid zygote. So the zygote in all of these life cycles or sex cycles, sexual life cycles that we're going to show, the zygote is always diploid. It's always 2N, okay? So it's a combination of 2 parents. Okay. Zygote obviously is then going to grow into a whole adult, just like you guys are. And then if you're female, you're going to produce eggs. If you're male, you're going to produce sperm. It's important that I tell you guys right now that for males, once you hit puberty, you're producing sperm every day, and always you're going to be producing 4 haploid cells. Females, humans, it's a little bit different, and the rest of female mammals, I think, as well, in that for female humans, you already have gone all the way through meiosis 1 for all the cells that are going to become eggs before you were born, when you were a fetus, which is like, whoa, mind blow. Right. And then once you hit puberty, one of those cells is going to finish meiosis 2. Okay. So let's take a real quick look at some of these other sexual cycles. And remember, for these, I'm going to be focusing mostly on definitions. So this is the sort of a simplified version. They put other animals here. We have a cute little mouse, right? Simplified version of the slide that I just showed you, right? So we've got the whole organism is diploid. It produces gametes. If it's male, it's going to produce male gametes. If it's female, it's going to produce female gametes. And then this is to try to show you that those 2 different gametes from different individuals are going to fuse to form a zygote 2N. And then we're going to start that cycle over again. It is a little bit different in plants, right? And it's called alternation of generations. And the reason it's called that is because some plants, some simple plants, which this is a moss here, they actually have 2 completely different organisms that produce the different either gametophyte, either gametes or spores. Flowers, which are an advanced plant or more evolved plant, they produce male and female parts inside one flower, right? So it's different for different kinds of plants. But what I'd like you to notice here, pay particular attention to, is let's start with the sporophyte down here. Okay, so this plant is diploid, the whole plant, and here this portion of it is diploid, which we call the sporophyte. And it helps me remember it if I think that sporophytes produce spores. And this is the part that's kind of backwards from us, in that the sporophyte itself is 2N. It's going to undergo meiosis to produce spores. The spores grow into a different organism, a multicellular organism, by mitosis. And this organism is called the gametophyte. See how it's just N? So it's only haploid. And see how it says mitosis, mitosis, right? The gametophyte is then going to produce gametes, and the gametophytes, sometimes they're on the same plant, sometimes they're separate. Usually, in this case, they're usually separate. So the one gametophyte is only going to produce sperm cells or pollen grains, and the other gametophyte is going to produce an ovule or egg cells. For most plants, they meet, those two different gametes are going to meet in water. Or if it's a angiosperm, like a flowering plant, in the wind or pollen, right? An insect might take pollen from one flower and take it to the other flower.
Okay.
Those are going to form a zygote, and then the zygote is going to form a sporophyte.
So it's like the zygote, the baby, really, if you want to say that?
Yes.
Like the product of everything
Yes.
coming together? Okay.
Yes. Yes. And then it's going to form an organism, but sometimes it looks like the other form, like the gametophyte, sometimes it doesn't, right? Try not to get too hung up on it because, like I said, I'm going to just talk to you. I'm going to ask on the test, I'm just going to ask the definitions of these words, okay? And then the last one, this applies to fungus, and it's very similar to the alternation of generations, but what is different about them, see how I wrote over here, the zygote is the only 2N form? You don't get multi, yes, you only get,
Hold up. What you want? I'm on a class right now.
I'm going to mute you. So this zygote is 2N, and it's only usually very short period of time. And then immediately the organism, the fungus, is going to go into meiosis, produce spores. And this is actually not a fungus. This is a protist. And the protists often look just, the two forms, the two gametes often look exactly alike. So you usually call them plus and minus rather than calling them male and female.
Okay.
Okay, so you'll see that later on. We showed this picture here because it's nice to see what a karyotype looks like of a human. So, and like I said, I'm on to page 2 of your notes. Nowadays, you can see this woman is performing the karyotype here. She's, you do it with a microscope, and she's ordering the chromosomes digitally, right? In olden days, we used to actually take a physical photograph through a microscope and then cut the chromosomes out and put them on a piece of paper. But now you can do it digitally. So this is actually a human karyotype, and it looks like it's male. See how it has an X and a Y? And this is a good time for me to remind you and to talk a little bit about, in humans, right, our diploid or adult form 2N equals 46, right?
Yeah.
N is 23. That means 23 different chromosomes that we have, right? And so in humans, if you're female, you're going to have a set of, we call it 45XX, because chromosomes are XX or XY, and in females, number 46 is XX. In human males, we would say 45, actually no, I think you actually say 46, sorry. And then we would say XY, and that would indicate that you're a male. Okay, and like I said, this karyotype shows an X and a Y. So this person would be a male. Notice they're ordered from largest to smallest is how you order a karyotype. So literally in all organisms, when you do one of these, the largest organism, the largest chromosome gets called chromosome 1. Simple as that. Now, this picture is super important for us to go over before we try to start talking about meiosis and all the phases of meiosis. And I think there's a lot, I know there's a lot of words on it, but if you learn some of this vocabulary back in chapter 12, it won't be so bad, okay? So I'm gonna walk you through this picture one step at a time. So the first thing I want you to look at is, do you see the blue chromosomes here? I think I can get it. Okay, that's too hard. All right, so the blue longest chromosome, so that would be chromosome 1, right? They're duplicated, so we still call them sister chromatids. And there's still a pinch, that's still the centromere, same. Now notice that they've made one pair of sister chromatids blue and one red. that are trying to show you that these are the chromatids that this organism would have gotten from its mother, and this one from, this one from the dad, this one from mom. Okay. Notice N equals 3 for this organism. So there are going to be 3 different sizes of chromosome. So we're going to call the biggest one chromosome 1, next largest chromosome 2, and the last one chromosome 3, the smallest one chromosome 3. Okay. Together, all 4 copies of chromosome 2 are called a homologous chromosomes or a homologous pair. Okay, and we didn't use that terminology. I didn't use that terminology in chapter 12 because in this chapter, we're going to see these homologous pairs all join together to form a tetrad in meiosis. They don't do that in mitosis. So, sort of an overall picture of meiosis. We start, so this is before. I like this picture, and I usually include this picture. I actually usually draw it out on the chalkboard in class with all 3 chromosomes in there, but this is a little simpler. So do you see this would be before, this would be during the cell cycle G1 of the cell cycle. So we've got
Just chilling.
Yep, we've got 2 chromosomes. If it was 46, if this was a human, there would be a total of 46 chromosomes, 2 copies of chromosome 1, 2 copies of chromosome 2, 2 copies of chromosome 3, etc. I think I mentioned it in the note outline. For myself, it helps me to remember when I'm looking at these blue and red chromosomes, if I think about the cells that are undergoing meiosis, if I think about them as my cells, and I think to myself, the blue one was from my mom, or the blue one was my chromosome 1 from my mom, and the red one is my chromosome 1 from my dad, right? So those are the ones that I inherited when I was a zygote. Now I'm a whole adult organism, and I'm going to produce gametes. So the first S phase is the same, right? So the cell cycle is always the same. G1, S, we duplicate all the DNA. Then we go into G2, and this time instead of going to mitosis, we're going to go to meiosis. So S, everybody gets duplicated, right? Now we call them sister chromatids. The first part of meiosis, we're going to separate the homologous pairs. See how I've got the blue ones are in one of the end result cells and the red ones are in the other cell, the end of meiosis 1. So then meiosis 2 is all about separating these 2 sister chromatids from each other, and these are the 4 gametes. And I usually draw them all as sperm cells because in humans, we always get 4 sperm cells. We don't always get 4 eggs. We only make 1 egg a month, right? But males make 4 sperm cells every time. So these would be haploid because they only have 1 copy of each chromosome. And that's good because when this sperm meets up with an egg, it's going to get a red chromosome, or it's going to get another chromosome 1 from the other parent when it forms a zygote. I'll give you a second to think about that. Or if anybody has a question about that.
So like in the interface right there, is this like, so can you put it in perspective of us now, like you were trying to do? Like the interface, the blue one is from my mom, let's just say now, and then the red one's from my dad.
Right, exactly. So this happened in you because you're female. It's Jada, right, that I'm talking to? Right? So this happened in you, this part at the end of meiosis 1, that happened when you were inside your mother, when you were a fetus.
Right.
Now, once you reach puberty, one of these cells, and it's either going to be this one or that one, not both, one of these cells is going to finish meiosis 2, and you're not going to get 2 resulting cells. You're only going to get 1 because we just make 1 egg as females.
Oh, so meiosis 1 happens while you're in the womb and meiosis 2 happens after.
Yes.
Okay.
Yes. Yeah. In women. In men, it's going on all the time, all the way through. 1, 2, 1, 2, 1, 2, their whole lives as soon as they reach puberty.
Okay.
Okay, so that's why I usually draw these as sperm cells because that's how it actually is in people. Okay, let's take a look at the phases real fast. okay? And you got to remember all of the things that are happening with the spindle fiber, the nucleus, and the chromosomes. Most of it is exactly the same as mitosis.
Mhm.
So if you memorize those three things from mitosis, this isn't so bad, because we're just adding a few new things, okay? All right, I'm gonna try to erase my board for a minute here. Okay, so let's take a quick look here at prophase 1. So again, cell cycle, after S, after G2, we're going into meiosis, prophase 1. Prophase 1 is probably the most different from mitosis. So I have the most to say about this, and then the rest of it's not going to be so bad, okay? So hang in with me for a few minutes here on this part. So let's take a look at this cell and what's actually happening to that cell. And I'm going to see if I can show you the blow up of that. So in prophase 1, notice, see how this chromosome 1 here, we've got both sister chromatids that were red and both sister chromatids that were blue.
Yeah.
Mhm.
They're all joining together. They join together, and when they're like that, we call them a tetrad, because it's all four sister chromatids together from one chromosome. Okay, so I'm going to draw that on the board real quickly here. So, try to make the cell really big, and I'm going to put prophase 1 here, and
You said it's called a tetrad?
it's called the tetrad. So I know the different colors of my chalk don't show up very well, so I'm going to, I'm going to draw one of these. These are all copies of chromosome 1, so red ones, blue ones, right? But I'm going to put diagonal lines through one of mine. Like I said, I know the colors don't show up that well. I'll try, I think green shows up kind of, right? So if I made these green, I don't know, I think that's too much. Let's just leave them plain and stripey, okay? Notice that I put a centromere in each one, and we would still call these each individually sister chromatids, sister chromatids. All four of them together, we call them a homologous pair, or when they're in prophase 1, we call them a tetrad. Now, the protein that your book is showing in between these to hold them together as a tetrad, I'm going to make it purple, because purple stands for protein in our book, always, right? And that protein has a special name, okay?
Oh wait, that protein is holding all four of them together, or is holding both pairs together?
Both pairs, both the pairs together.
Okay, okay.
And that protein's name is, it's more than one protein, so they call it the synaptonemal complex.
Somebody said, what the hell?
That's because another name for when the chromosomes cross over is synapsis.
Oh, okay. I know that word.
They're calling it the synaptonemal complex to help you think about it. Synapsis, SIS, because it's a process.
Yeah.
So like I said, there's lots of vocabulary, but you have to, try to sort it all out.
And that's just the many different proteins that's holding that pair together?
Holding this pair together. And it's holding all, so in these cells that they're showing in your book, N equals 3. So there's going to be 3 sets of tetrads, right? If we look back at the PowerPoint here, see how there's one set here, one set of tetrad here, one here. And this would be chromosome 1 because it's the largest, chromosome 2 because it's the second largest, and chromosome 3 because it's the smallest.
Okay.
Right? Now, when this forms, it provides the occasion for what we call crossing over or recombination. It doesn't happen on every single chromosome. It happens more frequently on the long ones. So the longer the chromosome is, the more likely you're going to have this crossing over event. And you that maybe watched my other video, right? So like if this, I've got my 4 pieces of string again, right? So this is my tetrad here for chromosome 1, and you can kind of see, even as I'm holding it, right, that these pieces don't always stay just with each other.
Right.
What actually can physically happen is one of my pink strands is crossing over one of the white strands. I twisted it around there. That's what crossing over means. There's the DNA, then the chromosome actually breaks and the pieces join back, but they don't always join back to the one they originally belonged to.
Is that how people get there are certain features and their,
Yes,
their
exactly.
stuff?
Exactly how that works.
Oh, okay.
So I'm going to draw a crossing over event just with this chromosome here.
Mhm.
So there's my plain chromosome 1, and here's my striped chromosome 1. And after a crossing over event, this plain chromosome 1 would get a piece maybe of striped, and striped gets a piece of plain. Right? And whatever characteristics are in this part of the chromosome, now those are the ones that are going to be passed on to your offspring.
Okay.
Okay? So this crossing over, so we call it crossing over because it literally looks like a crossing over. It's also called recombination. This only happens in prophase 1. Only happens in prophase 1. All capitals.
Go to the bathroom. Ask Dom to turn the light on for you.
Only happens in prophase 1. Now, one more vocabulary word or 2. After this is finished, as the cell moves from prophase 1 to metaphase 1, the synaptonemal complex disintegrates, but the last place that it disintegrates is where there was a crossover event. I'm going to draw that purple protein right there.
Yeah, I'm sorry, what was that? You said disintegrate?
Right, so those proteins, the synaptonemal proteins that are holding the tetrad together, as the cell moves from prophase to metaphase,
okay,
those start to disintegrate, but the last place they disintegrate is where there was a crossover event. So you can actually see it with a microscope.
Oh.
It looks like an X, right? So that's why they draw it that way in the pictures, right? That has a name, of course, right? That is called a chiasma, or the plural, chiasmata.
Too much.
You said it disintegrates at the last place that it crossed over?
Crosses over.
Yes.
Okay.
So that's the physical mark. Like when you're looking at cells under the microscope, you could actually see these under the microscope.
It literally says
Wait, what was that called again? That last going into metaphase 1, what was that called?
It's called a chiasmata. Right? It shows the physical crossing over of the two of the chromatids.
Okay.
Now, then the rest of it's very similar to we had in mitosis. See how the chromosomes are all lining up on the metaphase? Metaphase 1, still have a kinetochore, still have spindle fibers attached. Anaphase, the spindle fibers are going to shorten. Now, here's key. Put a star here. Anaphase, homologous pairs of chromosomes separate.
Say it again.
Homologous pairs of chromosomes separate. See how they're still
homologous pairs.
Together? 2 red ones together? Right? Telophase, same old. And where's the second part of this? Prophase 2, no crossing over this time. Metaphase 2, they've just changed the way they're drawing it. They're putting the metaphase chromosomes now horizontally instead of across the middle. Anaphase 2, now see how they're individual chromatids now separate? And they highlight that for you. Notice chromatids separate. And then we are going to end up with 4 cells that each cell only gets 1 copy of each of the chromosomes. So this particular cell here got a recombined chromosome 1, a non-recombined chromosome 2, and chromosome 3 that was also recombined. And as you guys pointed out, now if this happens to be the sperm cell that fertilizes the egg, you're going to get a mix of the chromatids that you had inside of you that you're passing on to your offspring. Right?
Who is playing something?
I don't know. I'm trying to go through and mute people but whatever. All right. So this is the summary picture. Okay. 1, 2, 3. See how there's 5 main properties or highlights or points for this, right?
Mm-hmm.
And they're comparing mitosis with meiosis. As I mentioned on the outline, this is usually the essay question.
Wait, which one? I'm sorry.
The
That's
difference between-
diagram.
Okay.
All right. I would expect you guys to be able to draw pictures or recognize mitosis
Draw a picture for what?
or meiosis. I would give you, I may draw pictures and ask you to identify the phases. And the best way for you to practice that, to get ready, is to draw it yourself.
Yeah.
Right? It's just practice. Draw it over and over.
So you should definitely have the quiz on Wednesday.
So what's the quiz gonna be on?
I'm gonna end it
with down,
guys. All right, but we can still talk. I wanna stop the video though before. I don't know how to do that.
And we can watch this video again in Zoom.
Yes, you can. So I will post the video. This video, I'll post it to YouTube first.
Okay.
And I'll put the link in your Canvas page so that when you open up Canvas in the module, I'll title it Video Meiosis. And you can click on it and then you can listen to the whole thing again.
Okay.
Okay? All right. Now, does anybody have any specific questions that you want to ask?
yes. Wednesday, what specifically should we know? Like, because this is difficult. I feel like we have to be in this class to like get it.
Yes.
I totally understand.
Yeah, it's online for
I
this.
made my video go away. That's all. I want this. Okay. I don't know how to turn off. Okay. Wait. Let me try one more thing here. More.